Hello everyone, let's get started with inflammation and healing. So, introduction, definition, and causes. Inflammation is defined as local response of a living mammalian tissue to injury from any agent. It is a body's defense reaction in order to eliminate or limit the spread of injurious agent, followed by the removal of necrosed cell and tissue. The injurious agent causing inflammation may be as under first infective agents like bacteria, virus, and their toxins, fungi, and parasites. Immunological agents like cell mediated and antigen mediated response. Third, physical agents like heat, cold, radiation, mechanical trauma. Fourth, chemical agents like organic and inorganic poisons. Fifth, inert materials such as foreign bodies. Thus, inflammation is a distinct from infection. Inflammation is a protective response by body to variety of etiological agent, infectious or non-infectious, while infection is an invasion into the body by harmful microbes and their resultant ill effects by toxin. Inflammation involves two basic processes with some overlapping wise early inflammatory response and later followed by healing. Though both these processes generally have protective role against injurious agent, inflammation and healing may cause considerable harm to the body as well. For example, anaphylaxis to bites by insects or reptiles, drugs, toxins, atherosclerosis, chronic rheumatoid arthritis, fibrous bands, and adhesion in intestinal obstruction. Immunity or immune reaction and inflammatory responses by host cell are both interlinked protective mechanism in the body. Inflammation is the visible response to the immune reaction, and activation of immune response is almost essential before inflammatory response appears. Signs of inflammation. The Roman writer Celsus in 1st century AD named the famous four cardinal signs of inflammation as rubor, redness, tumor, swelling, calor, heat, dolor, pain. To this fifth sign, functio laesa, loss of function, was later added by virtue. The word inflammation means burning. This nomenclature had its origin in older times, but now we know that burning is only one of the features of inflammation. Types of inflammation. Depending upon the defense capacity of the host and duration of response, inflammation can be classified as acute and chronic. A. Acute inflammation is of short duration, lasting less than 2 weeks, and represents the early body reaction and resolves quickly, and is usually followed by healing. The main features of acute inflammation are accumulation of fluid and plasma at the affected site, intravascular activation of platelets, polymorphonuclear neutrophils as inflammatory cells. Sometimes the acute inflammatory response may be quite severe and is termed as fulminant acute inflammation. B. Chronic inflammation is of longer duration and occurs after delay, either after the causative agent of acute inflammation persists for a long time, or the stimulus is such that it induces chronic inflammation from the beginning. A variant chronic active inflammation is the type of chronic inflammation in which during the course of disease there are acute exacerbations of the activity. The characteristic feature of chronic inflammation is presence of chronic inflammatory cell such as lymphocyte, plasma cells and macrophage, granulation, tissue formation, and in specific situation as granulomatous inflammation. In some instances, the term subacute inflammation is used for the state of inflammation between acute and chronic. Introduction to inflammation. Inflammation is a local response of living mammalian tissue to injury from any agent, which could be microbial, immunological, physical, or chemical agent. Cardinal signs of inflammation are redness, swelling, heat, pain, and loss of function. Inflammation is of two types: acute when due to early response by the body and is of short duration, and chronic when it is for longer duration and occurs after the delay and is characterized by response by chronic inflammatory cells. Acute inflammatory response by the host to any agent is a continuous process, but for the purpose of discussion, it can be divided into the following two events: vascular events, cellular events. Intimately linked to these two processes is the release of mediators of acute inflammation, which is also discussed just afterwards. First, vascular events. Alteration in the microvasculature, arterioles, capillaries, and venules is the earliest response to cell injury. These alterations include hemodynamic changes and change in the vascular permeability. Hemodynamic changes. The earliest feature of inflammatory response result from change in the vascular flow and caliber of small blood vessels in the injured tissue. The sequence of these changes is as under. Irrespective of the type of cell injury, immediate vascular response is of transient vasoconstriction of arteriole with mild form of injury. The blood flow may be re-established in 3 to 5 seconds, while with more severe injury, the vasoconstriction may last for about 3 minutes. Next follows persistent progressive vasodilation, which involves mainly the arterioles, but to the lesser affect other components of microcirculation like venules and capillaries. So note that persistent progressive vasodilation will be affect mainly arterioles and not the capillaries and venules. This change is obvious within half an hour of injury. Vasodilation results in increased blood volume in the microvascular bed of the area, which is responsible for redness and warmth at the site of acute inflammation. Progressive vasodilation in turn may elevate the local hydrostatic pressure, resulting in transduction of fluid into the extracellular space. It is responsible for swelling at the local site of acute inflammation. Slowing or stasis of microcirculation follows, which causes increased concentration of red cells and thus raised blood viscosity. 5. Stasis or slowing is followed by leukocytic mygination or peripheral orientation of leukocytes, mainly neutrophils, along the vascular endothelium. The leukocytes stick to the vascular endothelium briefly and then move and marginate through the gaps between the endothelial cells into the extravascular space. This process is known as emigration. Triple response. The feature of hemodynamic change in inflammation is best demonstrated by Lewis experiment. Lewis experiment induced the change in the skin of inner aspect of the forearm by firm stroking with the blunt point. The reaction so elicited is known as triple response or red line response, consisting of the following: red line flare wheel, red line flare and wheel, epidermis dermis pain nerve, triple response elicited by firm stroking of skin of forearm with a pencil, diagrammatic view of microscopic feature of triple response of the skin. First red line appears within a few seconds after the stroke and is due to local vasodilation of capillaries and venules. Second is flare, is bright reddish appearance or flush surrounding the red line and results from vasodilation of the adjacent arterioles. Third, wheal is the swelling or edema of the following skin occurring due to transduction of the fluid into the extravascular space. The features thus elicit the classical signs of inflammation, redness, heat, and swelling. Is due to fourth feature, pain has been added. So, what is a red line? Red line is the more presence of blood due to vasodilation of capillaries and venules. Flare is the reddish appearance or flush due to vasodilation. Red line is redness. Flare is the reddish appearance due to vasodilation, and wheal is the swelling or edema of the surrounding tissues. Now let's see altered vascular permeability. Pathogenesis. In and around the inflamed tissue, there is accumulation of edema fluid in the interstitial compartment, which comes from blood plasma by its escape through the endothelial wall of peripheral vascular bed. In the initial stage, the escape of fluid is due to vasodilation and consequent elevation of hydrostatic pressure. This is transudate in nature, but subsequently the characteristic inflammatory edema exudate appears by increased vascular permeability of microcirculation. The differences between transudate and exudate is summarized in table 4.1. Differences between transudate and exudate. Definition. Transudate is filtration of blood plasma without changes in endothelial permeability, whereas exudate is edema of inflamed tissue associated with increased vascular permeability. Character. Transudate is non-inflammatory edema, whereas exudate is inflammatory edema. Protein content in transudate is low, less than 1 gram per deciliter. Mainly albumin, low fibrinogen, has no tendency to coagulate. Whereas in the exudate, there is high protein content of 2.5 to 3.5 gram per deciliter, readily coagulates due to high content of fibrinogen and other coagulant factors. Glucose content of the transudate is same as in plasma, whereas in exudate it is low, less than 60 mg per deciliter. Specific gravity of transudate is low, less than 1.015, whereas of the exudate is high, more than 1.018. pH of transudate is greater than 7.3, whereas of the exudate is lesser than 7.3. LDH of transudate is low, whereas of exudate is high. Effusion LDH or serum LDH ratio is less than 0.6 for transudate and greater than 0.6 for exudate. Cells. Transudate, few cells, mainly mesothelial cells and cellular debris. In case of exudate, many cells, inflammatory as well as parenchymal. Example of transudate, edema in congestive heart failure, whereas in exudate, purulent exudate such as pus. Now let's continue. The appearance of inflammatory edema due to increased vascular permeability of vascular bed is explained on the basis of Starling hypothesis. According to this, normally the fluid balance is maintained by two opposing sets of forces. Force that cause outward movement of the fluid from the microcirculation. These are intravascular hydrostatic pressure and colloid osmotic pressure of interstitial fluid. Force that cause inward movement of interstitial fluid into the circulation. These are intravascular colloid osmotic pressure and hydrostatic pressure of interstitial fluid. Whatever little fluid is left in the interstitial compartment is drained away by lymphatics and thus no edema results normally. So areolar and where the hydrostatic pressure is 32 mm of Hg and osmotic pressure is 25 mm of Hg. So there is exclusion of the fluid into the interstitium due to the pressure difference. And later there is inclusion of the fluid at the venular end. There is no edema here, but here the hydrostatic pressure is normal and osmotic pressure is less. That is in case of vasodilation. So the osmotic pressure in the interstitium is increased, which leads to edema, which occurs in acute inflammation. Fluid interchange between the blood and extracellular fluid. However, in the inflamed tissue, the endothelial lining of microvasculature becomes more leaky. Consequently, the pressure of the interstitial fluid increases, resulting in excessive outward flow of the fluid into the interstitial compartment, which is exudative inflammatory edema. Early stage exemption, transudate vasculation, late stage exemption fluid, exudate increased vascular permeability. Patterns of increased vascular permeability. Increased vascular permeability in acute inflammation, by which normally non-permeable endothelial layer of macrovasculature becomes leaky, can have following patterns and mechanism which may be acting singly or more often in combination. Gap due to contraction of endothelial cells, contraction or mild endothelial cell damage, direct injury, endothelial injury by polymorpho-neutrophils, leakiness of neovascularization, arteriolar and venular and normal endothelial layer, interstitial edema, basement membrane. Schematic illustration of pathogenesis of increased vascular permeability in acute inflammation. The serial number in the figure corresponds to 5 number described in the text. So let's see. First is contraction of endothelial cells. This is most common mechanism of increased leakiness that affects venules exclusively, while capillaries and arterioles remains unaffected. The endothelial cells develop temporary gaps between them due to their contraction, resulting in vascular leakiness. This mediated by a release of histamine, bradykinin, and other chemical mediators. The response begins immediately after the injury and is usually reversible and is for short duration, 15 to 30 minutes. An example of such immediate transient response is mild thermal injury of skin of fora. Second is contraction or mild endothelial damage. In this mechanism, there is structural reorganization of the cytoskeletal of the endothelial cells that causes reversible retraction at the intercellular junction or mild form of endothelial damage. This change affects venules and capillaries and is mediated by cytokines such as interleukin 1 and tumor necrosis factor alpha. The onset of response occurs after the delay of 4 to 6 hours following the injury and lasts for several hours to days. Classical example of delayed and prolonged leakage is appearance of sunburns mediated by ultraviolet radiations. Third is the direct injury to endothelial cells. Direct injury to the endothelium causes cell necrosis and appearance of physical gaps at the site of detached endothelial cells. The process of thrombosis involving platelets and fibrin is initiated at the site of damaged endothelial cells. The change affects all the level of microvasculature, venules, capillaries and arterioles. So in the direct injury there occurs necrosis and appearance of physical gaps. So these gaps appears uniformly in all of the 3 vasculatures. Process of thrombosis involving platelets and fibrin is initiated at the site of damaged endothelial cells. The increased permeability may either appear immediately after the injury and last for several hours or days, immediate sustained leakage, or may occur after a delay of 2 to 12 hours and last for hours or days, delayed prolonged leakage. Examples of immediate sustained leakage are severe bacterial infections, while delayed prolonged leakage may occur following the moderate thermal injury and radiations. Fourth is leukocyte-mediated endothelial injury. Adherence of leukocyte to the endothelium at the site of inflammation may result in activation of leukocytes. The activated leukocyte release proteolytic enzymes and toxic oxygen species, which may cause endothelial injury and increased vascular leakiness. This form of increased vascular leakiness affects many venules and is a late response. The examples are seen in the sites where leukocytes adhere to the vascular endothelium, for example in pulmonary venules and capillaries. Fifth, leakiness in neovascularization. In addition, the newly formed capillaries under the influence of vascular endothelium growth factor VEGF during the process of repair and in tumors are exclusively leaky. These mechanisms are summarized in this table. Mechanism: first, endothelial cell contraction. The microvasculature involved is venules. Response type is immediate transient, lasts for 15 to 30 minutes. Pathogenesis involves histamine, bradykinins, and others. Example: mild thermal injury. Second is contraction or mild endothelial injury, which involves venules and capillaries, somewhat delayed in 4 to 6 hours, prolonged for 24 hours. Pathogenesis is interleukin 1 and tumor necrosis factor alpha. Example: sunburns. Direct endothelial cell injury occurs in arterioles, venules, and capillaries. Immediate, prolonged hours to days, or delayed 2 to 12 hours and prolonged hours to days. Cell necrosis and detachment occurs. Example is moderate to severe burns, severe bacterial infection, and radiation injury. Leukocyte-mediated endothelial injury occurs in venules and capillaries, delayed and prolonged response, leukocyte activation, and pulmonary venules and capillaries. Neovascularization occurs at all levels. Response can be of any type. Pathogenesis is angiogenesis and VEGF. Example: healing and tumors. Now let's see cellular events. The cellular phase of inflammation consists of two processes: exudation of leukocytes and phagocytosis. Exudation of leukocytes. The escape of leukocytes from the lumen of microvasculature into the interstitial fluid is the most important feature of inflammatory response. In acute inflammation, polymorphonuclear leukocytes comprises the first line of body defense, followed later by monocytes and macrophages. The changes leading to migration of leukocytes is as follows. First, normal axial flow, then margination and pavementing, then rolling and adhesion, then immigration and diapediasis. Sequence of changing in the exudate of leukocytes: A. Normal axial flow of the blood with central column of cells and peripheral zone of cell-free plasma. B. Margination and pavementing of neutrophils into the narrow plasmatic zone. C is adhesion of the neutrophils to endothelial cells with pseudopods in the intracellular junctions. D is emigration of neutrophils and diapediasis with damaged basement membrane. First, change in the formed elements of the blood. In the early stage of inflammation, the rate of flow of the blood is increased due to vasodilation, but subsequently there is slowing or stasis of the bloodstream. With stasis, change in the normal axial flow of the blood in the microcirculation takes place. The normal axial flow consists of central stream of cells comprised by leukocytes and RBCs and peripheral cell-free layer of plasma close to vessel's wall. Due to slowing and stasis, the central stream of cell widens and peripheral plasma zone becomes narrower because of loss of plasma by exudation. This phenomenon is known as margination. So, what is margination? Due to slowing and stasis of the central stream of cells widens and peripheral plasma zones becomes narrower because of the loss of plasma by exudation. As a result of this redistribution, neutrophils of the central column comes close to the vessel wall. This is known as pavementing. Second, rolling and adhesion. Peripheral marginated and pavemented neutrophils slowly roll over the endothelial transient. Bond between leukocytes and endothelial cell become former, that is adhesion phase. The following cell adhesion molecules bring about rolling and adhesion phases. First is selecting. These are a group of cell adhesion molecules as expressed on the surface of activated endothelial cells and are structurally composed of lecithins, or lectin-like protein molecules, the most important of which is S-Lewis X molecule. Their role is to recognize and bind to glycoprotein and glycolipid of the cell surface of neutrophils. There are three types of selectins: P-selectin, E-selectin and L-selectin. P-selectin performed and stored in endothelial cells and are platelets, also called as CD62, is involved in rolling. E-selectin, synthesized by cytokine activated endothelial cells, also named as ECMP, is associated with both rolling and adhesion. L-selectin expressed on the surface of lymphocytes and neutrophils and are called as LCAM, is responsible for homing of circulating lymphocytes into the endothelial cells in lymph node. P-selectin performed and stored in endothelial cells and platelets, is involved in rolling, whereas E-selectin, synthesized by cytokine activated endothelial cells, associated with rolling as well as adhesion, and L-selectin for homing of circulating lymphocytes to the endothelial cells. Second is integrins. These are the family of endothelial cell surface protein having alpha CD11 and beta CD18 subunits, which are activated during the process of loose and transient adhesion between endothelial cells and leukocytes. At the same time, the receptors for integrins on the neutrophils are also stimulated. This process brings about form adhesion between leukocytes and endothelium. Third is immunoglobulin gene superfamily adhesion molecules. This group consists of a variety of immunoglobulin molecules present on most cells of the body. The partake in cell-to-cell contact through various other CAMs and cytokines. They have a major role in recognition and binding of immunocompetent cell as under cellular adhesion molecules ICAM1, also called CD54, and vascular cell adhesion molecule VCAM1, also named as CD106, allow a tighter adhesion and stabilize the interaction between leukocytes and endothelial cells. Platelet endothelial cell adhesion molecule 1, PECAM or CD31, is involved in leukocyte migration from the endothelial surface. Third is emigration. After sticking on neutrophil to endothelium, the former moves along the endothelial surface till the suitable site between the endothelial cell is formed, where the neutrophil throw out cytoplasmic pseudopods. Subsequently, the neutrophils lodged between the endothelial cells and the damaging it locally with secreted collagenases and escape out into the intravascular space. This is known as emigration. The damaged basement membrane is repaired almost immediately, as already mentioned. Neutrophils are the dominant cells in acute inflammatory exudate in the first 24 hours, and monocyte macrophage appear in the next 24 to 48 hours. However, neutrophils are short-lived 24 to 48 hours, while monocyte macrophages survive much longer. Stimulus to emigration of leukocytes, escape of red cells through the gaps between the endothelial cells, diapidiasis takes place. It is a passive phenomenon where RBC is being forced out either by raised hydrostatic pressure or may escape through the endothelial defect left after emigration of leukocytes. Diapidiasis gives hemorrhagic appearance to the inflammatory exudate. Chemotaxis, transmigration of leukocytes after crossing several barriers, that is endothelium basement membrane, perivascular myofibroblasts and matrix to reach the interstitial tissue, is a chemotactic factor mediated process called chemotaxis. The concept of chemotaxis is well illustrated by Boyden's chamber experiment. In this millipore filter of 3 micrometer pore size separate the suspension of leukocytes from the test solution in the tissue culture chamber. If the test solution contains chemotactic agent, the leukocyte migrate through the pores of the filter towards the chemotactic agent. Polymorphonuclear neutrophils, PMNs, test solution, chemotactic agent. So this is chemotactic agent and this is which is the test solution, and these are neutrophils. And there is the micropore. So as the chemotactic agent attracts the neutrophils, so there is a establishment of chemotactic chemical gradient which leads to the flow of neutrophils towards it. So this is the figure of Boyden's chamber with micropore filter shown by dotted line. Suspension of leukocytes above is separated from the test solution below. Lower half chamber shows margination of neutrophil towards the chemotactic agent. The following agent act as potent chemotactic substance. For neutrophils, leukotrienes B4, LTB4, a product of lipooxygenase pathway of arachidonic acid metabolites. Second is component of complement system C5a and C3a, in particular cytokines, interleukins, in particular IL-8, soluble bacterial product such as formylated peptides to responds and partake in inflammation having specific chemokines, for example monocyte chemoattractant protein MCP1, eotaxin chemotactic for eosinophils, NK cells for recognizing virally infected cell, etc. Phagocytosis. Phagocytosis is defined as the process of engulfment of solid particulate material by the cells, cell eating. The cells performing this function are called as phagocytes. There are two main types of phagocytes: polymorphonuclear neutrophils, which appear early in the acute inflammation response, sometimes such as macrophages. Second is circulating monocyte and fixed tissue mononuclear phagocytes, commonly called as macrophages. The neutrophils and macrophages, on reaching the tissue space, produce several proteolytic enzymes: lysozymes, protease, collagenase, elastase, lipase, proteinase, gelatinase, and acid hydrolase. These enzymes degrade the collagen and extracellular matrix. The phagocytosis of the microbe by polymorphs and macrophage involve in three steps: recognition and attachment, engulfment, and killing and degradation. Stages of phagocytosis of foreign particles. A is opsonization of the particle. B is pseudopod engulfment of the opsonized particle. C is incorporation within the cell phagocytic vacuole and degranulation. And D is phagolysosome formation after fusion of lysosome to the cell. IgG opsonin, Fc receptor, lectin, C3b receptor, C3b opsonin, bacteria, pseudopod, degranulation, phagocytic vacuole, and phagolysosome formation. Recognition and attachment. Phagocytosis is initiated by expression of cell surface receptor on macrophage, which recognize microorganism, Mannose receptor and scavenger receptor. The process of phagocytosis is further enhanced when the microorganisms are coated with specific protein, opsonins, from the serum, and the process is called opsonization, meaning preparing for eating. Opsonins establish a bond between bacteria and cell membrane of phagocytic cell. The main opsonins present in the serum and their corresponding receptor on the surface of phagocytic cell, PMNs or macrophages, are as under. IgG opsonin is the Fc fragment of immunoglobulin G. It is the naturally occurring antibody in the serum that coats the bacteria, while the PMNs possesses the receptor for the same. C3b opsonin is the fragment generated by activation of complement pathway. It is strongly chemotactic for attracting PMNs to bacteria. Lectins are carbohydrate binding proteins in the plasma which binds to bacterial cell wall. Engulfment. The opsonized particle or microbe bound to the surface of the phagocyte is ready to be engulfed. This is accomplished by formation of cytoplasmic pseudopods around the particulate due to activation of actin filament beneath the cell wall enveloping it in phagocytic vacuole. Eventually, plasma membrane enclosing the particle breaks from the cell surface so that the membrane-like phagocytic vacuole or phagocyte becomes internalized. So the plasma membrane enclosing the particle will break from the cell surface to internalize the material in the cell and lies free in the cell plasma. The phagosome fuses with the cell or more lysosome of the cell and forms bigger vacuole called phagolysosome. Then final killing and degradation. Next comes the stage of killing and degradation of microorganism to dispose it off, which is the major function of phagocytes as scavenger cells. The microorganism after being killed by antibacterial substance are degraded by hydrolytic enzymes. However, this mechanism failed to kill and degrade some other bacteria like tubercular bacilli. In general, following mechanisms are involved in disposal of microorganism: intracellular mechanism, oxidative bacterial mechanism by oxygen free radicals, MPO dependent, MPO independent, oxidative bactericidal mechanism by lysosomal granules, non-oxidative bactericidal mechanism, and extracellular mechanism. These mechanisms are as follows: intracellular mechanism. Intracellular metabolite pathway are involved in killing microbes, more commonly by oxidative mechanism and less often by non-oxidative pathway. Oxidative bactericidal mechanism for oxygen-free radicals, an important mechanism for microbial killing, is by oxidative damage by the production of reactive oxygen metabolites O2 superoxide, H2O2, OH, HOCl, HOI, HOBr. A phase of increased oxygen consumption, respiratory burst by activated phagocytic leukocyte, requires the essential presence of NADPH oxidase. NADPH oxidase present in the cell membrane of phagosome reduces oxygen to superoxide. 2-oxygen is reduced to 2 superoxide anions by NADPH oxidase. Superoxide is subsequently converted to hydrogen peroxide, which has bactericidal properties. This type of bactericidal activities is carried out either via enzyme myeloperoxidase MPO present in the azurophilic granules of neutrophils and monocytes, or independent of the enzymes MPO, as under A. MPO dependent killing. In this mechanism, the enzyme MPO acts on hydrogen peroxide in the presence of halides, chloride, iodide, or bromide to form hypohalous acid. This is called hydrogen peroxide MPO halide system and is more potent antibacterial system in polymorphs than hydrogen peroxide alone. B. MPO independent killing. Mature macrophages lack the enzyme MPO and they carry out bactericidal activity by producing hydroxide ions and superoxide singlet oxygen from hydrogen peroxide in the presence of superoxide Haber-Weiss reaction or in the presence of Ferrous-Fenton reaction. Hydrogen peroxide Haber-Weiss reaction in the presence of superoxide produces hydroxide, and in the presence of ferric ion Fenton reaction produces hydroxyl radical. Reactive oxygen metabolites are particularly useful in eliminating microbial organism that grows within the phagocytes. For example, M. tuberculosis, Histoplasma capsulatum. Second, oxidative bactericidal mechanism by lysosomes. Granules in this mechanism perform granule stored product of neutrophils and macrophages are discharged or secreted into the neutrophils and macrophage are discharged or secreted into the phagosome and the extracellular environment. While the role of MPO is already highlighted above, other substances liberated by degranulation of macrophages and neutrophils are protease, trypsinase, phospholipase, and alkaline phosphatase. Progressive degranulation of neutrophil and macrophage along with oxygen free radical degrades the protein, that is, induce proteolysis. Third is non-oxidative bactericidal mechanism. Some antigen released from the granules of phagocytic cells do not require oxygen for bactericidal activity. These include A granules. Some of liberated lysosomal granules do not kill by oxidative damage but cause lysis of microbes within the phagosome. These are lysosomal hydrolysis, permeability increasing factor, cationic proteins, defensins, lipases, proteases, and DNases. B is nitric oxide. Nitric oxide is a reactive free radical similar to oxygen reactive radicals, which is formed by nitric oxide synthase. It is produced by endothelial cell as well as activated macrophage. Nitric oxide is another potent mechanism of microbial cleaning. Extracellular mechanism. Following mechanisms explain the bactericidal activity of extracellular levels. Granules. Degranulation of macrophages and neutrophils explained above continues to exert its effect of proteolysis outside the cell as well. Immune mechanism. As already discussed in chapter 3, immune mediated lysis of microbes takes place outside the cell by mechanism of cytolysis, antibody mediated lysis, and by cell mediated cytotoxicity. Acute inflammatory response. The sequential hemodynamic changes are an initial transient vasoconstriction followed by persistent progressive vasodilation, raised local hydrostatic pressure, and transudation in extracellular space. Next, there is emigration of leukocytes from the capillary wall due to slowing and stasis in microcirculation. There is increased vascular permeability and appearance of inflammatory edema. It has different mechanism and various patterns. Contraction of endothelial cells, mild to severe endothelial damage, direct or leukocyte mediated injury to endothelial cells, and leakiness of neovascularization. Cellular phase of inflammation consists of exudation of leukocytes and phagocytosis. Leukocyte exudation begins from change of normal axial blood flow to slowing and stasis. This is followed by margination, pavementing, rolling, adhesion, and finally emigration of leukocytes to the extravascular space. Phagocytosis is cellular eating, the process of engulfment of foreign particulate material involvement into the initial recognition and opsonization. The mechanism of phagocytosis are largely intercellular, oxidative and non-oxidative, and few extracellular mechanisms.